Szanowni członkowie komisji, drodzy uczestnicy i słuchacze, szanowni państwo. W imieniu zarządu Studenckiego Towarzystwa Naukowego Śląskiego Uniwersytetu Medycznego w Katowicach chciałabym serdecznie podziękować za przybycie i uczestnictwo w konferencji International Medical Congress of Silesia SIMC 2024. Nazywam się Julia Herbuś i mam przyjemność koordynować sesję nauk podstawowych, biotechnologii i inżynierii biomedycznej. Serdecznie witam Komisję Naukową, w której skład wchodzą doktor nauk medycznych Anna Kleczka, która jest z nami zdalnie.
Dzień dobry Państwu.
Magister Aleksandra Lasia oraz pan magister Marcel Madej.
Dzień dobry.
Rozpocznę od kilku informacji organizacyjnych. Jeżeli chodzi o kolejność prac, pierwszą pracą będzie praca o tytule Morphological Analysis of Ganglia Spinala ze względów technicznych. Jest to praca, która w książce abstraktu znajdowała się jako ósma. Reszta prac będzie przedstawiona wedle kolejności w książce abstraktów. Czas przeznaczony na prezentację wynosi 7 minut. Następnie po prezentacji będzie się odbywać 3-minutowa dyskusja. W przypadku przekroczenia czasu uczestnik zostanie przez nas poinformowany i poproszony o przejście do wniosków i zakończenia prezentacji. I tutaj jeszcze mam kilka informacji dla komisji. Przypominam, że jeżeli praca prezentowana jest, mają Państwo ją pod opieką, to nie mogą Państwo tej pracy oceniać. Reszta komitetu normalnie może taką pracę ocenić. Jeżeli to jest praca po prostu z katedry, też może podlegać ocenie przez Państwa. I jeżeli praca byłaby prezentowana w języku angielskim, to pragnę przypomnieć uczestnikom, że na pytania, nawet zadane w języku polskim, należy odpowiedzieć w języku angielskim. Jeżeli chodzi o ocenianie prac, to oceniana jest wartość merytoryczna w skali od 1 do 5, sposób prezentacji w skali od 1 do 5 oraz sposób i forma prowadzenia dyskusji w skali od 1 do 5 i na koniec twórczy wkład własny w skali od 1 do 5. Po zakończeniu sesji od komisji stacjonarnej zbiorę karty tutaj, a zdalnie automatycznie prześlę się do naszego systemu. Jeżeli nie ma żadnych pytań od uczestników, myślę, że możemy zaczynać. Dlatego prosiłabym o udostępnienie ekranu przez pana Marka Misiaka jako osobę, która będzie prezentować jako pierwsza.
Dzień dobry. Czy jestem słyszalny?
Tak, słyszymy pana. Proszę tylko o udostępnienie całego ekranu, nie tylko okna PowerPointa.
Oczywiście. Czy teraz jest prezentacja dostrzegalna przez Państwa?
Jak najbardziej widzimy prezentację.
Jak rozumiem, mogę rozpoczynać, prawda?
Oczywiście, jeżeli pan jest gotowy, można rozpocząć prezentację.
Good morning, ladies and gentlemen. Thank you for possibility of presenting my results from my research. Today I'm going to speak about ganglia spinalia, and this is the agenda for our meeting today with main points that I'm going to focus on during the whole presentation. So to start, I would like to define the term of ganglia spinalia. I think that we all remember this structure from our anatomy course from the first year of medical studies. So what is most important is that it's a structure that consists of primary sensory neurons, and it is on a track of the posterior spinal roots, obviously near the place where ventral and dorsal roots join all together, making the spinal nerve. Our purpose was to clarify the normal morphologic features and variations of lumbar ganglia spinalia in healthy population. So I have carried out microanatomic dissections of lumbar spinal nerves from L1 to L5. As a biological material, we had 8 formalin-fixed human cadavers, and I used Zeiss surgical microscope. So now I will bring down some anatomy for you. So in every book that we may read, I mean neurosurgery books, anatomy books, anesthesiology, or just any textbook from the topic of spinal surgery, there is illustration or just chapter focusing only on ganglia spinalia with illustrations, but in each of those books we have only one model, one variation of ganglia spinalia. It's a monoganglion, so it is only one ganglial component. It obviously has this characteristic fusiform or just spindle-like shape, and it's extremely strange because in a real human body, we obviously can find monoganglia, but there is also possible to find biganglia and triganglia as well. So depending on how many ganglial components, maybe I will try to show you that, depending on how many ganglial components there are, we can differentiate biganglia and triganglia. And between those ganglial components, there might be obviously a very thin layer of connective tissue. I have found only 4 articles covering the topic of ganglia spinalia in different ways, mostly location or just architecture of connecting patterns. But in fact, polyganglia, so biganglia and triganglia, were only mentioned. Nobody really focused on their morphology or just variations. So I tried to focus on this topic. That's what those authors shown in their articles. We had monoganglia, biganglia, so 2 ganglial components that are seemingly independent, and they have also focused on MRI pictures. So we've got 3 ganglia in here with 3 seemingly independent ganglial components. But the thing isn't as easy as it may seem. In here, we've got those seemingly independent ganglial components, but sometimes we can also find biganglia with incomplete division. So there is a thin layer between those ganglial components, but from one side, it is like only monoganglion. But looking from the other side, we've got a groove or the sulcus for ventral root of spinal nerve. And according to literature, we can say that monoganglia can be found most often at most levels. Biganglia, even in half of the cases, on the level of L3 and L4. There was unfortunately no data about the level of L5. Triganglia at the same time could be found only occasionally at the levels of L2 and L5. According to my own experience, I can confirm that from L1 to L5, monoganglia can be found most oftenly. But biganglia, well, in here, we've got numbers that go down. So at the level of L3, we've got only 18%. At the level of L4 and L5, 25%. And triganglia can be found occasionally at the level of L4 and even up to 20% at the level of L5. I would like to emphasize that ganglia spinalia is a very important structure in mechanisms of many disorders like sciatica, radicular symptoms, also obviously low back pain. So if we want to prepare the correct treatment for patients, we have to understand the proper anatomy of ganglia spinalia. At the same time, it is very necessary for spine surgeons to understand this architecture as they want to minimize the incidence of injury of ganglia spinalia during different operations. Obviously, future studies should explore the existence of functionally distinct biganglia because that may inform the placement of the electrode lead for ganglia spinalia or just neurostimulation. The presence of biganglia and triganglia may affect the accuracy and quality of electrical stimulation of ganglia spinalia. And still we do not know if the distribution of dermatomal innervation in polyganglia is the same as in monoganglia. This is my bibliography. Thank you very much for your attention. And if there are any inquiries, I would be happy to answer all them. Thank you very much.
Bardzo dziękujemy za prezentację i teraz zachęcam komisję, jak i uczestników do zadawania pytań.
Can I start? Thank you for the presentation, but I have a few questions. Could you please tell me, do you have any information about the patients from whom you have the slices from the analysis?
Unfortunately, such information are not available because I was working in a research club of anatomical studies, so it was quite hard to get such information.
But do you have any confirmation that they were healthy individuals without any changes? Yes, yes.
Yeah, because
but
the reason of death was never the problems with neuronal cell bodies from ganglia spinalia.
Sorry that I'm interrupting you. But you don't have any information about the age of the patients, yes, their sex, for example?
Well, actually, such information wasn't really necessary for us.
Necessary.
Yeah, because it doesn't change, according to the literature, anything. Like if somebody is born with polyganglia in any level, it will stay till the death.
Yes. I understand. So the anatomy is not connected with the age, yes, or the sex. But don't you think that having only 8, yes, slides is not enough to have such a conclusion?
Well,
would you like to broaden your yes project?
Obviously, my project will be broadened in the future. But for this moment, it is enough because our hypothesis was to show that in textbooks, there is only shown one morphology variation, and still in the human body, we can find much more variations. So for this hypothesis, to confirm it, it's just enough. We just confirm that it is possible to find biganglia, triganglia, and so on, and different other morphology. And if we want to ensure that we have every kind of variation, obviously it will have to be in the future broadened so that we can have much bigger biological material.
Especially because you proved before that there is a lack of literature data, yes, about this type of analysis, yes. Thank you so much. I have no other question.
Thank you very much.
Czy są jeszcze jakieś pytania z sali? W takim razie, jeżeli nie ma pytań, kończymy dyskusję. Bardzo dziękujemy za prezentację, jak i za całą dyskusję. I zapraszam do zaprezentowania kolejnej pracy. Design and construction of a robot feeding a patient with paralysis of the upper limbs. I to też jest praca zdalna, więc proszę o udostępnienie ekranu z panią Agnieszkę.
Przepraszam, czy ja mogłabym mieć tylko taką pragmatyczną uwagę, zapytanie?
Tak,
czy mogłabym prosić, żeby po każdej prezentacji zostawiono nam taką minutę, przynajmniej dla mnie potrzebną na wypełnienie formularza oceny?
Oczywiście.
Dobra, 30 sekund, minuta, żebym po prostu sobie poklikała w tym formularzu oceny, jeżeli to nie byłoby nadużyciem. Dzięki.
To prosiłabym panią Agnieszkę, żeby przed prezentacją jeszcze pozwoliła, chwilkę zaczekała, żeby pani doktor...
Dobra, już. Dziękuję.
Dobrze, w takim razie możemy zaczynać.
Tak, słuchać mnie dobrze?
Troszkę cicho, nie wiem jak zdalnie pani doktor.
Halo, czy mnie słychać?
Tak, w porządku.
Dobrze, a więc zaczynam. My name is Agnieszka Adamczyk, and the topic of my presentation is design and construction of a robot feeding patient with a paralysis of the upper limbs. This project was supervised by Professor Katowice, Professor Detyna. As the human population continued to age, there is a growing need to develop new tools for assistance tools who are dependent on caregivers. Specifically, individuals suffering from Parkinson's disease experience difficulties in coordinating movement that hinder independent meal consumption, particularly with liquids. People after stroke are also in need of assistance, and the most important group, individuals with motor disabilities who cannot live independently. It is crucial to popularize and make accessible those solutions to everyone who needs them. To assist caregivers in their responsibilities, I developed a feeding robot as a part of my master's thesis. The project aimed to design and construct a competitive feeding robot. While there are existing feeding stations available in store, as demonstrated in the presentation, they are often not affordable for everyone. My robot was primarily created using additive manufacturing method, 3D printing, and is controlled by an Arduino Uno. As a result, the final station price should be competitive, and the model itself should be easy to reproduce at home and affordable to manufacture. The slides show one of the robot's components, designed and ready for printing using Ultimate Cura software. And here we can see the diagram of the electronic parts. Beside Arduino, we can see, among others, 4 servo mechanism, RGB LEDs, a card reader, and temperature sensor. The card reader is an innovative addition that allows patient data to be stored on a special card, optimizing robot arm movement for individual patient. The temperature sensor is integrated in the ball handle is also innovative addiction, enable control of the meal's temperature. And here we can see the whole model of the manipulator and the physical prototype version. And here we can see the whole station. The feeding station includes a robot, a bowl, a spoon, a card reader, a temperature sensor, an RGB LED, main switch, and control buttons. They are control buttons are here enlarged. And this first one is for height changer. This another one is for making break from eating or finish using. And the last one is for taking another portion of food. Now, meanwhile, all electronics, including battery, are enclosed with a casing, providing protection against accidental damage. The station was tested on a food product of various consistencies and successfully fulfilled its function. A video recording of the whole working robot is also included. Here we can see the process of preparation of the food, including placing the bowl in the holder and positioning the spoon in the gripper. At the beginning, LED is red because station is not ready yet. Now we are tapping the cards and we can see station is ready. Now diode is red because food is hot. And robot scoops the food into the spoon. Checks this to remove the excess and serves it to the user. We can see the LED turn to yellow because the temperature drops. Here, user is changing the height of the spoon and asking for another portion of food. So now we can see robot taking another portion. This is a soup, and we will see robot handling food of different consistencies as well. can see the picture. It's almost here. Now we can see the cold shell. This is more solid. And finally, robot return to the resting position, and user will turn it off. Conclusion. The project demonstrated that creating a competitive priceless feeding stations entirely feasible and hold great potential for the future. The test confirmed that the station fulfills its role and can be produced using relatively available materials, making it an affordable option for those in need. However, to move the prototype out of the laboratory and commercialize it, adjustment would be necessary to improve the robot's design and ensure accessibility for people with disabilities. Here's the sources, and thank you for your attention. Any questions?
Bardzo dziękujemy za prezentację. Tutaj prośba oddziału technicznego. W momencie dyskusji prosimy o wyłączenie prezentacji, żeby były lepiej widoczne osoby zdalnie uczestniczące. I zapraszam komisję, jak i uczestników do zadawania pytań.
Does anyone have any questions, or can I just ask something?
Na razie z sali nie ma pytań.
Okay, so thank you for your presentation, and I'm amazed by the device you've constructed, and I know that is crucial to lower the price of such a robot, but I do think that if it have to be still improved, yes, this device, yes, and I'd like to ask you, what do you think about the relation between patient and the robot? Because I'm thinking that people who are ill, yes, who don't need any help, will better cooperate with other human being, yes? And how do you think people will be cooperate with the robot, yes? Especially the elderly one.
That's dependent of individuals needed. There are people that are a little embarrassed if someone have to help them with food. So thanks to robots, they can feel independent.
That's the truth, but you've presented that preparing the food, yes, and preparing the robot for its work, you have to be involved in that processes, yes? You have to just put the food into the microwave, yes, pull it into special frame. So do the patients will have the ability to do that, or they need some extra help?
They need extra help, but then when food is served, everyone can just sit in the table and eat together. Thanks to this, a whole family can enjoy the meal together in the same moment.
That is very important, yes. And I would like to stress that we will be in high need, yes, of such a devices in the future, yes. Thank you so much. Thank you.
Thank you.
Okay, I also have a question at first. Do you hear me well? Or not?
Yes, I'm hearing. I'm hearing you.
I just would like to ask you, what was your own contribution into this project, from idea to the end of the project and testing the robot?
It was my idea from the very beginning. I started this, and the full process was almost only my job. Of course, I have my supervisor to help me and with advice, but the whole process was almost just my job. The only things I didn't was able to make myself was the 3D printing because I don't own any. So it was a full job of my supervisors.
Okay, thank you so much.
And can I ask another question? Maybe I didn't hear it, but how much will your robot cost?
We don't know yet, but
Approximately.
I guess it could be 1,000-2,000. So it's much.
It's quite approachable for the patients.
Yeah.
Yes. Thank you.
Bardzo dziękujemy za prezentację i całą dyskusję. Teraz sekundka na uzupełnienie kart oceny dla komisji.
Dziękuję.
Czy komisja stacjonarna też już gotowa? Dobrze. W takim razie zapraszam kolejną osobę, tym razem już stacjonarnie, prezentującą pracę Human Body in the Dissection Room. What do you think about it?
Dear committee, honorable guests, my name is Martyna Rożek, and today I would like you to present the results of my and my co-author's work titled Human Body in the Dissection Room. What do you think about it? And the work has been prepared by the students from the Science Club from the Department of the Anatomy and the Department of the General and Vascular Surgery, both from the Medical University of Silesia in Katowice. And our work tutor is Dr. Tomasz Lepich from the Anatomy Department. And the main aim of our work was to find out about the significance of using the human body in the process of anatomy learning, also learning and the awareness and the opinions of students of the donation of the human bodies, and also finding out about the concerns for the body donation. And for our study, we used an original single choice questionnaire that has been prepared especially for the needs of our study. And it contains 20 questions and has been answered by 528 students from medical universities from all over the Poland. 366 of them were women and 162 of them were men. The median age was 21 years old, and most of the respondents were from the first year of medical studies. And 57%, which is more than half, identified as Catholics. When it comes to our results in terms of the body and organ donation willingness, only 16% of the respondents claimed that they would like to donate their body for the educational purposes before starting the medical course. And after that, after starting the medical course, the number rose in a percentage to the 35% for the willingness of the body donation. And what is statistically significant here, that more Catholics are willing to donate their bodies than more atheists are willing to donate their body than Catholics. And it is 36.9% to 26.8%, which is really significant. And when it comes to the organ donation, here you have the very big contrast, because 91% of medical students are willing to donate their organs for the body transplantation. And also, it is not statistically significant, but also atheists are willing more to donate the organs than the Catholics. And here is something that's really important, that females are more willing to donate their organs for the body transplantation, but males are more willing to donate their body for the educational purposes. And if we want to compare men and women willingness to donate their bodies for educational purpose, in our study, the number of males that are willing to do so is above 30%, 40%. And it is actually really similar and comparable to the works from all over the world. We have studied many of them. And it's generally a fact that males are more willing to donate their body for scientific purpose. But here in the last row, you have one of the really few works from all over the world that shows that females are more willing to do so. And here in the last row is an example of a work from India. Here we have a table that showed the number of the students that are considering donating. And if we put a line on the 50% and said that everything above 50 is that students are generally speaking willing to donate or considering the donation, and everything that's below 50 is that generally speaking students do not want to do so, we are on a level of 35%, which is generally speaking very similar to the works from all over the world. But here you have, just as an information, there are also some exceptions from all over the world, and there are a few works. And here in the last bottom two rows, you can see there are two works where you can see that the number of students that are considering donating is higher than 60%. And how does medical studies affect the willingness to donate their body? First and foremost, more than half of the participants, it's 57%, have encountered an act of disrespect for donor bodies during anatomy class. And this is the main source for the concerns for body donation. So as I said, the concerns are shown here on the chart, and the fear of the other students disrespecting the body is their biggest concern. 30% claimed that they have no concerns whatsoever. Then there were psychological considerations, objections of the family, and also religious considerations. And because of the act of the disrespect the students have encountered, it is generally said that they will not encourage their family members to donate their bodies for educational purposes. Here you have that the number of the students that will not do so is at 72.5%. But what is really interesting that they are more willing to encourage the people that they are not related with, that they are really not know well, to donate their body for educational purposes. But how we can change the students' approach towards the human bodies? So most of the students believe that it's really might be useful to receive more information about the body donors, that it might be gaining more respect for the human bodies. Also, 65% believe that getting to know the representatives of the family members before even starting the anatomy course would increase the students' respect for the donor bodies. And that gives us a clue what we can do, what we can change. So the anatomy lesson can not only be about learning the body structures, the human anatomy, but also can be a very important lesson of the respect that is much needed in the future doctor's career. And we also asked our respondents about the significance of the human bodies in anatomy classes, and only one person claimed that they have not used the human bodies in their anatomy lessons. And what is really interesting, 94% of the students believe that it is not possible to teach anatomy efficiently and effectively in medical school without the access to the real human bodies. And even in the era of technological development, almost 90% of the respondents believe that there is no alternative teaching method that can replace the real human body's anatomy teaching. And when we are coming up to the conclusions, we wanted to raise the awareness about the significance of using the human bodies in the anatomy learning, that it is a very useful tool. And right now in Poland, our study showed that there is no better method and no better tools that can replace human bodies. And the main issue right now is that there are not so many people that are willing to donate their bodies for the educational purposes. And right now, by finding out why medical students, people that are almost today on the first year of medical studies, in contact with the real human bodies, why they don't want to do it, why they will even discourage their family members not to do so, we were able to identify the issue. And right now we can solve it. We can do something to change it, to gain more respect for the human bodies. So the long and hard process of the anatomy learning can be a very fruitful source of the knowledge, not only about the body structure, but something that is the most fundamental subject in a medical and future doctor's career, and the hard lessons of the respect. Thank you for your attention. I am open to the discussion.
Okay, thank you for your presentation. It was really great. I think that your topic about awareness of donation is really neat, especially now. And I would like to ask I'm really curious, what pushed you into examination of this topic?
Basically, from our own experience, we also have encountered the fact that sometimes there are not so many human bodies in the dissection room. Also from our passion for our participation in the scientific club from the anatomy department, we see there's the issue that there are not so many bodies. Then we see the state that the bodies are in. Sometimes they're very old, they're not very useful for anatomy learning. And also we have witnessed the acts of the disrespect and the anatomy lessons in the anatomy classes. And we think that is a serious issue. And by changing that, solving that, we can really maybe even revolutionize the anatomy learning process.
Okay, thank you.
Can I? To be honest, I'm terrified by the statistics that over more than half of medical students witness acts of disrespect of the bodies. And I'm curious, what do you mean about disrespect? How students treated the body?
So first, the thing that is very common, there are taking pictures of the human bodies, which is really against the rules. Then there are some acts when that's in professional acts of, I don't know, taking the parts of the human bodies, showing them, making fun of them. It's very unprofessional, and we see how the students are sometimes laughing. They're not taking the subject seriously, and we think it's a big issue because it's their first contact with the human body. We know that it's technically dead, right? But it still needs the big amount of the respect, the same as the real living human being. And if that will not be taught in the first year of their studies, maybe they will not learn it during the later on medical course. And that's not the type of the doctors we want to raise at this university.
Yes, I agree that it is crucial to be respectful to patients, but don't you think that we should also do something to just teach the student that they have to act properly in the dissection room rather than trying to convince the body donors that they will be fine after the death?
Yes.
How can we just I don't know, how can we prove to the students that they are having contact with the body, with the human?
So there were some of the examples that we showed in a questionnaire, like getting to know the family members, getting more information about the donors. But we believe that, and we want to implement something like this in our future anatomy courses, that before even starting the anatomy course, that the first lesson should not be bones or something, the structures. It should be something very brief, something very short, but something that will give
something very obvious, to be honest, yes?
Something that will give them a second just to think about the significance of the not thing that they are doing right now, but the thing that they will be doing for the future. Something that is really important in the future job. And I don't know how if it will work, but we can try, and that's the only thing we can do.
Yes, I think that it is crucial just to stress that the human is the human even after the death. Yes, the death. Sorry. Thank you. Thank you so much. The topic is very interesting.
Dziękujemy bardzo za prezentację i za całą dyskusję. Teraz czas na wypełnienie kart ocen przez komisję.
Jasne. Dziękuję.
Dobrze, dziękuję i zapraszam do prezentacji następnej pracy The Role of the c-KIT Receptor in the Regulation of Autocrine Growth of Bladder Cancer Cell Lines. I tutaj ta prezentacja będzie zdalnie, więc proszę o udostępnienie ekranu.
Good morning, ladies and gentlemen. I hope you can see me and hear me. I'm right?
Everything is fine, yes.
Thank you. My name is Patrick Obajtek, and I have the pleasure of presenting a work entitled The Role of the c-KIT Receptor in the Regulation of Autocrine Growth of Bladder Cancer Cell Lines. So, let's start. Okay. Bladder cancer ranks as the second most frequently diagnosed cancer within the genitourinary system across the Europe. It is known for its frequent reappearance and carries a substantial risk of advancing a more severe stage. Studies show that the increased activation of receptor tyrosine kinases, for example, a c-KIT, may play a significant role for the development of this type of cancer. What to say? Furthermore, current research show that the invasion and progression of this cancer may be closely associated with overexpression of receptor tyrosine kinase activity, such as c-KIT or PDGFR. And for these reasons, tyrosine kinase inhibitors, for example, dovitinib, are among the most advanced examples of personalized medicine. The aim of the study was to assess the impact of the selected tyrosine kinase inhibitor, dovitinib, on a selected bladder cancer cell lines. The study utilized bladder cancer cells from a different level of malignancy as a research material. And on this slide, you can see the mechanism of action of dovitinib. And on the other slide, you can see various inhibitors of the tyrosine kinase receptor, like dasatinib or sorafenib. And you can see their sites of the action. So what's about the methods? Surface antigen CD117 was labeled using suitable monoclonal antibodies attached to either phycoerythrin or fluorescein through specific staining methods, and subsequent analysis was carried out utilizing a flow cytometry. The growth rate of cancer cells after inhibition application was assessed in vitro using a proliferation assay with crystal violet and the MTS assay. Additionally, the impact of inhibitors on cell viability was assessed using flow cytometry with annexin 5 and propium iodide staining. What's more, the potential effect of dovitinib on the migration ability of cancer cells was assessed using a scratch assay. The immunocytometric analysis revealed the expression of the c-KIT receptor on all examined cell lines. And using the MTS assay, it was demonstrated that the inhibitor inhibits the proliferation of all tested cell lines in concentration and time-dependent manner. Based on the MTS test, the inhibitory concentration values were determined, and for the RT4 cell line, it was approximately 8 micromoles for the test compound, while for the T24 cell lines, it was approximately 13 micromoles. What's more, FACS analysis showed that the tested compounds primarily induce apoptosis in cancer cells. So, based on the scratch assay test, inhibition of cancer cell migration was observed under the influence of the inhibitor. So make a short conclusion. Dovitinib represents a promising compound for potential targeted therapy of bladder cancer. And what's more, due to the involvement of tyrosine kinases in tumorigenic process, the search for new specific inhibitors of these enzymes with clinical significance appears desirable. So that's all from me. Thank you very much for your attention.
Bardzo dziękujemy za prezentację i zapraszam do dyskusji.
I have a question. Cell lines. Do you know the differences between these two cell lines?
Yes, the RT4 cell line is less harmful for the organism, when the T24 cell line is more dangerous. It's grow very fast, and it's very difficult to treat that cancer.
Okay, and do you know about differences in the gene mutation which may influence to the effect of Dovitinib? Because
sorry, could you?
Some lines may have a gene mutation corresponding to the difference responses to Dovitinib. So my question is, how these lines do this line have such a mutations, or just this not was a case of the study?
This is not a case of the study. We testing Dovitinib on this two bladder cancer cell lines. So we don't know how it can change the work and the action of Dovitinib if we have some mutation in these lines.
Okay, thank you.
Thank you very much.
It may be crucial, yes, what was said before. I'd like to ask you if you know any other methods of treating the bladder cancer others than chemotherapy.
Other ways of chemotherapy? Yes, so in the nowadays oncology, we have so many treating ways. The first way is the chirurgical resection of the bladder cancer, but it is very harmful to the patient. So we are trying to use some new methods like targeted therapy, and nowadays we have some compounds that can do it, and we have some drugs to make this way. But
Sorry. What about the immunotherapy?
I think in this time we don't have any immunotherapy in the normal guidelines to treat this cancer. But I think this is the future of the oncology, and soon it can appear some drugs for it. So the FDA and the United States is the leader of this science. But in Poland, we don't have at all on this time any new drugs to cure this cancer.
But what about BCG vaccination, which is sometimes put into the therapy of bladder cancer patients and is connected with the activation of immune system? I've heard not a long time ago that it is used, especially in oncological places such as Kielce Hospital, for example. It is widely used, to be honest. But it was not a subject of this presentation, I know. I have only one more question. Could you tell me what kind of device did you use in the scratch test? What was the device used?
It was the cells was on the plate, and we scratch it by I don't know how to say it. It's a metal pen, and we scratch it, And next, under the microscope, we was checking on the time how much time it takes to the cell family claims together.
But do you have any device for these? Something like Julibert, for example, or you just put the slide under the microscope?
Yeah, it's full manual. It's full manual.
Thank you so much. Thank you for the answers.
Thank you.
Bardzo dziękuję za prezentację i za dyskusję. Teraz chwila dla naszej komisji, żeby uzupełniła karty ocen.
Dziękuję. Ja dziękuję.
Komisja stacjonarna też już gotowa? Okej, w takim razie zapraszam osobę prezentującą tą kolejną pracę o tytule Reliability of the Heart Rate Variability for Assessing the Automatic Nervous System in Healthy Volunteers.
Okay, so am I audible? Am I visible?
Yes, I can hear you very well. Thank you.
Yeah, so good morning. First, I would like to warmly welcome all participants of our conference. My name is Damian. I am a student of Medical Informatics at Wrocław University of Science Technology, further known as Politechnika Wrocławska in Polish. Besides studies, I work with the Pondicherry University, which is a new university in India, as a co-author of a desktop software application, which is a master thesis of a student in India. I'm also sorry that I am not in a physical form on the conference, but due to the illness, I couldn't get there. Also, I hope that my illness won't interrupt me in my speech. And so, going to the study. This study was done with Miko Ainaida, and the work tutor was a PhD advisor, Agnieszka Uryga, who's working in Brain Lab in Politechnika Wrocławska. Yes. So today I would like to present to you a topic of HRF matrix analysis, dependency of a time window it was measured in. So first, let me talk about finance and agreements. So this study was financed from a grant called Sonata 18, issued by NSC, which stands for National Science Center. Also, study was approved by Bioethical Committee of Wrocław Science and Technology. So what I'm going to present and why we are even interested in that. So heart rate variability is a measure of variation in a time intervals between heartbeats. It is regulated by autonomic nervous system. And for the motivation, it would be that comparability and readability of HRF signal duration have not been previously accessed. So now I'm going to talk about the aim of the study, but first let me explain what you can see on the side of the slide. So basically, this is a plot of NeuroKit to Python library. This is a programming analysis, so kind of programming information will be put in there. And basically, this is a ACG signal, as you can see here, measured by a mean of signals. So further down, I will talk about the NeuroKit library later, but here you can see the perfectly visible ACG signal, which is not always the case. So study aims to investigate the impact of an ACG signal measuring duration on three different HRF metrics: RMSSD, ratio, LF, and HF, which is low frequency and high frequency, and sample. These all metrics are three different metrics of HRF metric. Yes. The study was conducted on healthy volunteers in the number of 15, and we are basically only interested in healthy patients. So patients with, for example, arrhythmia were excluded from the analysis. And obviously they are very visible in this ACG signal because it is, the ACG is not visible with the patient who has an arrhythmia. So materials and methods. This slide would be covered very, very briefly. So here we can see the stuff which is which made the study possible, basically. This is a measuring device. It is a photoplethysmograph, Finan Presnova from FMS Medical Systems. And it was measured at rest during a 30 minutes period. So HRF parameters were estimated using Python, as I said, with RHF and NeuroKit2 libraries. And 4 different time periods were selected. So 3, 5, 10, and 13 minutes were selected throughout the 15 healthy volunteers with average of age 23 plus minus. And here we can see the not obviously whole, but a part of a Python script, part of which I was working on. And this is also ICC, interclass correlation coefficient equation, which was necessary to further compare the data. Equation is kind of self-explanatory, so I would not go into details about it. I could obviously explain it in the discussion. So here we have the results, and the results suggest that analysis revealed no significant differences in RMSSD between time periods, but meaningful differences were found for LF and HF ratio as well for the sample. And here we can see the full results. So here the 1 stands for 3 minutes, 2 for 5, 3 for 10, and 4 for 30 minutes. So as we can see, this is our mean values of all the analysis, and they are not vary too much between other time periods. So the conclusion would be that this study suggests that ACG signals with a measurement time greater than 3 minutes do not significantly influence HRF metrics. This might indicate that a 3-minute measurement duration might be significant enough to justify HRF metrics. And further research is needed to explore the implications of these findings because the ICC revealed meaningful differences between sample and LF-HF ratio. So why this is important? This is important because, for example, for my next study, which I'm also doing for Brain Lab, I needed to analyze not 15, but 100 volunteers. And basically this, for example, with 30 minutes time period, it would take roughly 2 days to analyze it with a software that I have written. But when we simplify it only to 3 minutes, it would take like 3 or 2 hours. So this extreme in time improvement and debugging, because debugging of a software that takes 2 days to run, it's a nightmare, I would say. So this would be it for a conclusion. Here you have the contact information of mine, as well as a GitHub repository in which the code is stored. This is my LinkedIn, this is my mail. If you would have a nice project, I am also very excited to contribute to it. So this is all for me, and I'm open to discussions.
Bardzo dziękuję za prezentację. Tutaj prośba tylko o jej wyłączenie na czas dyskusji, żeby było lepiej widać. Właśnie, dziękuję. I zapraszam komisję do dyskusji.
I have one question about your own contribution to this project.
Yes, so I have written the whole software basically. Like I have written the whole analysis. If you want to know more about tech details, I can explain it like right now. Maybe not right now because they are kind of like very sophisticated, but yes, it would not be a problem.
Thank you.
I have to admit that your work was very difficult to understand for me because I'm not such a technical girl. But I'd like to ask you about what do you mean telling us that you have healthy volunteers? What was your excluded, included, yes, conditions? Yes.
So as I presented, there was a ACG signal presented there, and basically this image is not visible at all when, for example, having a patient with arrhythmia, and we had that in our in our analysis, but this needed to be excluded because then if you would add this person to our analysis, the differences would become like extreme. It might even get to 1,000 higher than normal ones. So basically, it may be even considered as a technique to determine if patient has an arrhythmia because it indicates that it is very, very different than normal ones.
But why have you chosen the volunteers specifically at the age of 23, for example?
They are considered as the most healthy ones. So basically, in my mind.
Of course, it is just a theory.
Yeah. So basically then, yeah.
What about the sex?
The sex
can
was
it
different.
influence? Yes, can it influence to the results of your work?
No, sorry.
Just take your time, of course.
No, the sex does not influence.
Okay.
Oh, sorry, my illness is taking the time.
Okay, so wishing you to be healthy. Thank you for your presentation.
Yeah, thank you as well.
Bardzo dziękujemy za prezentację i za dyskusję. Teraz chwila dla naszej komisji.
Dziękuję.
Dziękuję bardzo i zapraszam autora kolejnej pracy Joint Symbolic Analysis of Blood Flow Velocity During Controlled Breathing in Healthy Volunteers. So our topic was joint symbolic analysis of blood flow velocity in healthy volunteers during controlled breeding. Okay, so we would like to express special thanks of gratitude to our tutor, Dr. Agnieszka Uryga. Ethical approval for this study was granted by Bioethical Committee of Wrocław Medical University, and this project is financed by grant from the Sonata National Center. Okay, so baroreflex is an autonomic mechanism which maintains blood flow to organs by keeping blood pressure steady. And baroreflex mediated relationship between arterial blood pressure and cerebral flow velocity is not well understood. So joint symbolic analysis is a method which could detect nonlinear relationships between ABP and FV. We wanted to discover whether joint symbolic analysis of ABP and FV time series could detect cardiovascular autonomic activity caused by controlled breeding in healthy volunteers. Okay, so 24 young healthy volunteers were breeding at 6, 10, at rates of 6, 10, and 15 breaths per minute, while we recorded blood flow velocity in middle cerebral artery with transcranial Doppler and arterial blood pressure with photoplethysmograph, and simultaneous recording of FV and ABP was possible thanks to ICM Plus software. Okay, so I computed heart rate from ABP time series using Fourier transform, and I extracted peak systolic velocity and interval time series from flow velocity and arterial blood pressure time series. And then I computed differences between subsequent values and transformed them into binary sequences. So if a difference was negative or equal to 0, then it indicated 0. And if the difference was positive, it indicated 1. And then I formed words of length 3 and put their frequencies into word density distribution matrix. So here is the exemplary word density distribution matrix, and I computed 2 metrics from these matrices. First one is JSD-SIM as a trace of this matrix, and JSD-DIAM as an anti-trace of this matrix. Okay, so lower breathing rate was associated with lower heart rate for 6 breaths per minute. Also, lower breathing rate was associated with lower JSD-SIM values for 6 breaths per minute. And there was a statistically significant rise in JSD-DIAM values for 6 breaths per minute. Okay, so reduced JSD-SIM values suggests reduced non-baroreflex activity. Significant rise in JSD-DIAM values suggests higher baroreflex patterns. And joint symbolic analysis captures baroreflex activity, and the study should be repeated in a larger group. Thank you. Dziękujemy za prezentację i zapraszam komisję do dyskusji.
Are there any questions, or can I?
Yes.
Thank you. Sorry.
Yeah, I would have a question. What libraries have you used to access this data?
Okay, so I used SciPy library to extract the PSV and error values.
Anyone else? As I've said previously, I am not good in such a technical works, but I am quite interested of how your research can be useful in normal life. Yes, how can we just have any any potential, yes, for using it in diagnosis of patients, for example?
Okay. So this project is a part of a bigger project, which aim is to find a link between baroreflex sensitivity and brain perfusion. And we want to understand this link to help traumatic brain injury patients.
Okay. And the patients who have suffered from some brain issues, yes, could be diagnosed and treated, yes, by the devices, by the ways you.
I think we would like to know faster that they will be having problems.
So it is about prevention.
Prevention, like at the moment of being admitted to hospital, before there is a sudden decrease in a patient health. We would like to know faster that they're going to have a problem.
So I would like to just check if I understand you correctly. Patients who are having some symptoms, yes, will be just diagnosed by your system, yes, to know earlier that they are having some issues, yes? No.
So it's not this point. So it's not. I don't think it's the step. I think it's a lot it's a start.
It is just a start, yes, of the research, yes?
Yeah.
You don't even know how useful it could be, yes? It have to be broadly investigated. Thank you.
It was just a start. We just wanted to check whether it would work. It would detect anything.
Yes, I appreciate that you are thinking about such a big thing. Yes, I understand. It is just the beginning of your research. Thank you.
Hey, thank you for presentation, of course. And as we said, we are not so technical, so I just would like to ask about your own contribution in this project.
Okay, so my job was to implement this algorithm in Python. I also wrote the abstract and made a presentation.
Okay, thank you.
Bardzo dziękujemy za prezentację i za dyskusję. Teraz chwila dla naszej komisji.
Dziękuję.
I zapraszam do prezentacji kolejnej pracy o tytule The Impact of the TCF7L2 RS 7903146 gene polymorphism on the carbohydrate metabolism.
Good morning, everyone. My name is Aleksandra Wiśniewska. I represent the Student Scientific Group Pharmacon at the Medical University of Warsaw. And today I would like to present our research about the impact of the TCF7L2 gene polymorphism on the carbohydrate metabolism. First, to start off with some words of introduction, TCF7L2 is short for transcription factor 7 like 2.
I'm sorry to interrupt you, but I cannot see the presentation. Okay, thank you. I just cannot see it.
Czy widzi pani doktor teraz ekran?
Tak, ale nie w formacie prezentacji, w sensie nie w... O właśnie, tak.
A teraz widać?
Bardzo dziękuję. Bardzo dziękuję. Przepraszam, że pani przerwałam.
Nie, bardzo dziękujemy za informację.
Okay, so I'm gonna start over. As I said, I represent the Student Scientific Group Pharmacon at the Medical University of Warsaw. And today I would like to present our research about the impact of the TCF7L2 gene polymorphism on the carbohydrate metabolism. And the TCF7L2 is short for transcription factor 7 like 2, and it is one of the main regulators of the WNT signaling pathway. This pathway is involved in processes such as cell migration and cell proliferation, and therefore it influences embryogenesis, cancerogenesis, and was also described as a main factor influencing pancreas function. And therefore, single nucleotide polymorphisms of the gene coding the TCF7L2 were proven to influence metabolic functions and might be related with elevated risk of various metabolic diseases. The aim of our study was to assess the impact of the TCF7L2 gene polymorphism on the prevalence and severity of carbohydrate metabolism dysfunction. We involved in our study 441 generally healthy patients who reported for control occupational checkups. And we performed DNA isolation, genotyping with the use of a real-time PCR method, biochemical analysis such as fasting glucose, total cholesterol, LDL, HDL, and triglycerides, and finally statistical analysis with the use of ANOVA and chi-squared test. The first part of our results show us the distribution of the genotypes and alleles in our study group. We can see that almost 60% of the group had the CC genotype, and it is the most common genotype, and 4.5% had the TT genotype. The allelic and genotypic distribution was consistent with the results that are available in online genetic databases. Here we can see the distribution of genotypes according to the occurrence of type 2 diabetes in our patients. We found significant connection between the occurrence of diabetes and the genotype of this polymorphism. And we can see that the prevalence of TT genotype was much higher in the group of patients with diabetes compared to healthy patients. We also examined the differences between genotypes considering fasting glucose concentration, and we found statistically significant differences with the TT genotype predisposing to the highest fasting glucose concentrations in our group. Because the carbohydrate metabolic dysfunction, especially type 2 diabetes, are diseases that commonly occur with other diseases, metabolic diseases such as obesity, hypertension, and dyslipidemia, we decided to expand our study and also include these factors. So here we can see that we found statistically significant connection between the occurrence of obesity and the genotype. And similarly to the previous slide, the prevalence of the TT genotype was much higher in the group of patients with obesity compared to patients with normal weight. Also we found that the TT genotype predisposed patients to higher BMI values compared to patients with normal weight. As I mentioned before, we also compared the distribution of the genotypes in patients with and without hypertension. However, we did not find a statistically significant connection. Also about the lipid profile, we did not find any statistically significant differences between total cholesterol, LDL, HDL, and triglycerides between our genotypes. So to sum up our results, we can tell that there were the carbohydrate metabolism dysfunction present in our group, and they were connected to the genotype of this polymorphism. This polymorphism was strongly associated with type 2 diabetes, mellitus, and obesity among our studied group. And the TT genotype predisposed to significantly higher fasting glucose concentration as well as BMI values. And that's all. Thank you very much for your attention.
Bardzo dziękujemy za prezentację i zachęcam komisję, jak i uczestników do zadawania pytań.
I have a question. After the extraction, did you perform a qualitative and quantitative analysis of the obtained extract?
Yes, we did assess the quality and quantity of our DNA that we obtained, and it was all according to the standards that we were given to have the best quality of our report.
Thank you.
Thank you very much.
Okay, thank you for your presentation. I have a question about real-time PCR. What do you use as a negative or positive control in your in this step of your experiment?
Yes, so we didn't have a positive control because we were genotyping the patients, so we didn't know about their genotype before we did the PCR. And for the negative control, we just used the mix for the PCR without any patient samples. So instead of the patient, there was water.
Thank you so much.
Thank you very much.
If I may, you've examined 441 patients. Yes, is it correct? How long does it take?
It's not as long as you would think, because when we are doing a PCR, the one plate has 96 wells, so we can already do 95 patients on one plate. So it wasn't that long. It was like a few weeks.
But what about constructing this group? How did you find the volunteers, yes, and people who are just fulfilling your included-excluded conditions?
So, as I mentioned, it was the group of patients that were on their occupational checkups. So it was in a collaboration with one of the clinics that can do these checkups. And we just asked the patients additionally for another consent if they want to participate in this research. And they provided us with information about their health, about their other diseases, and their serum samples for the biochemical analysis.
Thank you. And I'd like to ask you, do you think that people who are having very specific polymorphism of the gene you were talking about should be, should be told to have some extra diet or some activities just to try to help them not to having such a condition as obesity, yes, or different glucose levels, for example?
I think that it is very important if the patient already knows that they might be at higher risk of developing at this point probably diabetes or obesity, that they are informed about this possibility. And we also have to remember that it's not like a sentence. It's not that if they have this genotype, they're gonna be sick. It's just some higher predisposability. So it would be important that they are informed and educated about the things they can do to stop this from happening. But it's mostly just, it's not any form of treatment because it's not a disease yet. However, any dietary requirements, any activity, physical activity is very much preferred.
Yes, because many patients are thinking that because they're having some kind of gene polymorphism, yes, they cannot change anything, yes, and they are acting sometimes very early. So it is crucial to stress that they can influence their state, yes?
Yes, exactly. Yeah, at this point, it's nothing is sure and we can still change it.
Thank you so much.
Thank you very much.
Just briefly about your own contribution, because I see that laboratory work, I think it took a lot of time, and then of course statistical analysis. So you do it everything on your own?
So we already had the biochemical analysis results because they were done at the clinic. However, all of the DNA isolation and the genotyping I did myself.
Thank you.
Thank you very much.
Bardzo dziękujemy za prezentację. Teraz chwila dla naszej komisji.
Dziękuję.
Okay, now I would like to ask to present the paper entitled Bioinformatics Study of the Deleterious Non-Synonymous SNVs in the human SHH gene. Anna, you are with us? Okay.
Yes. Good today, dear jury and everyone present at the meeting. My name is Anna Ipatova. Allow me to present to your attention a report on this topic, bioinformatic analysis of pathogenic and putatively pathogenic single nucleotide variants of the human SHH gene. The SHH gene encodes the sonic hedgehog signaling protein, which plays a pivotal role in human embryotic and post-embryotic development, especially in the development of the central nerves and the digestive system, eyes, limbs, etc. Mutations in the SHH gene caused several birth defects, including non-syndromic holoprosencephaly, agenesis of the corpus callosum, and microphthalmia. Despite the fact that disease susceptibility in humans is frequently associated with single nucleotide variants, SNVs, and evolutionary conserved genes frequently contain disease-associated SNVs, there was no systematic analysis of SNVs in the human SHH gene. The aim of the research was to evaluate in silico the function implication of non-synonymous SNVs in the SHH gene. Materials and methods include 411 non-synonymous SNVs of gene SHH were retrieved from UniProt and ProtVar database. Total number of deleterious non-synonymous SNVs in SHH gene was estimated using CADD tool. The potential effects of SNVs on the protein structure and functions were predicted by 8 bioinformatic tools with different algorithms: MAPP, PenSer, PhD, SNP, polyphen-1, polyphen-2 predict SNP, CIFT, and SNAP. Protein stability was evaluated using IMUTAN, MUPRO, and iStable predicts. CADD is combined annotation depletion dependent depletion is a web-based tool that provides a detailed analysis of variants along with a Phreat score value that generally predicts the probability of a variant to induce deleterious effect on the protein. Exon coordinates of SHA gene were uploaded on the CADD, and data on 4,194 potential SNVs were obtained. For 2,775 of them, Phreat score was equal or greater than 15, thus they were categorized as deleterious. Only 119 of those SNVs were labeled as pathogenic in the bioinformatic basis. Then we applied 8 bioinformatic predicts of potential effects on SNVs on the protein structure and functions. MAPP is an online tool based on physicochemical property and conformity assessment. It identified 183 deleterious SNVs. Spencer estimated position as specific evolutionary preservation of amino acid. It labeled 239 SNVs as probably pathogenic or Spencer estimated position-specific evolutionary presentation of amino acid. It labeled 239 SNVs as probably pathogenic or possible domating. PhD SNP uses a combination of support vector machine trained on a defect sequence and evolutionary information to predict variant pathogenicity. It detected 179 deleterious variants of SHA gene. Polyphen-1 predicts possible impact of an amino acid substitution on the structure and function of a human protein using straightforward physical and comparative constitution. It found 196 deleterious SNVs. Polyphen-2 is an updated edition of the previous tool. It detected 255 deleterious SNVs of SHA gene. Predict SNP is a concise classifier of prediction mutation associated with disease. It indicates 175 deleterious variants of SHA gene. CIFT is a sequence homology-based tool that predicts amino acid substitution caused by SNVs and their subsequent impact on protein function and pathogenicity. It found 194 deleterious SNVs. SNAP is a tool based on neural network analysis of simple single nucleotide substitution. It identified 162 deleterious SNVs. Thus, a minimum number of deleterious SNVs were detected by SNAP, while the larger one was found by Polyphen-2. Difference in the number of gene variants identified as pathogenic by different bioinformatics services are likely due to the peculiarities of their algorithms. Next, an analysis of the pathogenicity of SNVs of the SHA gene was carried out based on the results of the assessment by all 8 services used. Variants were labeled as likely pathogenic if they were identified as deleterious but by at least 4 reserves and labeled as pathogenic if they were identified by at least 6 bioinformatics tools. It was detected 168 pathogenic and 24 likely pathogenic SNVs for SHA gene. However, 87 pathogenic and likely pathogenic SNVs were found in the bioinformatics basis. 152 deleterious SNVs were not mentioned there as pathogenic. SNVs may cause not only alternation of the structure and function of the protein, but may affect its stability too. 3 bioinformatics predicts of protein stability were applied: I mutant, MU pro, and I stable. Negative short mean that protein stability was discrete. It was found that 301 SNVs decrease protein stability. 143 out of them were pathogenic and likely pathogenic, and 220 of them were newly detected that confirmed our evolution of SNV's pathogenicity. So on the slide, you can see that conclusions that have already been announced during the report. Thank you for your attention.
Thank you for your presentation. Now I would like to invite you all for question and discussion the present paper. Sorry.
If I may, your work was very theoretical, and I'd like to ask you if you see any opportunities to use your research in clinic. I mean, could your research be useful, for example, in the prenatal diagnosis of some diseases?
Yes, this research can be used in prenatal genetic diagnosis of embryos for artificial insemination.
Okay, and could you tell me the very specific role of the sonic Hedgehog signaling protein in the embryotic processes? What this protein does? Sorry.
The protein plays a pivotal role in development of the central nervous and digestive system and musculoskeletal system and nervous system.
So in many different processes, yes, it is involved, this special protein. Thank you so much.
Thank you.
Thank you so much. I teraz będzie chwila dla komisji.
Dziękuję.
Dziękuję bardzo za informację. Czy komisja stacjonarnie też już gotowa? Dobrze. Czy jest na sali autor pracy Assessment of Psychological and Metabolic Parameters after the Implementation of CGM in DM1? Dobrze, w takim razie, jeżeli uczestnik dołączy do nas, to jego prezentacja będzie zaprezentowana na koniec. A teraz wedle kolejności zapraszam autora pracy Effect of Cafeteria Diet and Intestinal transportation surgery on glucose metabolism in SPD rats.
So I can also start. Good morning, ladies and gentlemen. My name is Kinga Pixa, and today I'm going to tell you about effect of cafeteria diet and intestinal transposition surgery on glucose metabolism in SPD rats, which I was working on with my colleagues. And we are from also Department of Physiology, Faculty of Medicine, Medical University in Silesia in Katowice. So I think now we can start. Obesity has become a disease that affects all social groups, moreover, has been declared an epidemic. There is increasing evidence to support the notion that regardless of procedure, bariatric or metabolic surgery is safe and effective to the treatment of obesity and type 2 diabetes mellitus. Now moving to the general skin of the... Czy ja mogę poprosić inną prezentację? Bo to jest chyba źle ekranie, bo ja mam inną. Sorry, we have little problems with this presentation because it's not the same as I sent it.
Prosimy o sekundkę przerwy, ponieważ mamy tutaj w takim razie problemy techniczne.
Proszę się niczym nie denerwować, takie sprawy się po prostu zdarzają.
Tak, oczywiście, tutaj za sekundę będziemy już mieli tą prawidłową prezentację, więc wszystko będzie w porządku. I tutaj też czas będzie jakby od początku naliczony, więc na spokojnie.
So let's begin again. Good morning, ladies and gentlemen. My name is Kinga Pixa, and today I'm going to tell you about the effect of cafeteria diet and intestinal transposition surgery on glucose metabolism in SPD rats, which I was working on with my colleagues, and we are from also Department of Physiology, Medical University of Silesia. So let's begin. Obesity has become a disease that affects all social groups, moreover has been declared an epidemic. There is an increasing evidence to support the notion that regardless of the bariatric or metabolic surgery is safe and effective in the treatment of obesity and also type 2 diabetes mellitus. Now moving to general scheme of the study. We selected 96 rats after 7 days of acclimatization. The rats underwent 12 weeks feeding protocol before the surgery. During this feeding protocol, we divided into two groups these rats. One group is control diet, and the second one is cafeteria diet, the green arrow, the first green arrow. Prior to the surgery, rats starved for 12 hours and then were randomly assigned to the second groups with IT and sham surgery. After the surgery, rats are divided into subgroups, which I describe later. So now I would like to start by drawing your attention to the figure 1. Ooh, not this. To the figure 1, which shows a male Sprague-Dawley rat. Also take a look at the figure 2. These rats were hosted in cages maintained at specific environmental conditions, like temperature ranging from 20 to 30 degrees Celsius, and also relative humidity between 50 and 70%. So now, next, please take a look at the following slide about the cafeteria diet. So cafeteria diet is kind of the Western diet. The diet lacked vegetables, lacked fruits and flower seeds, and also cereals. With this in mind, let's proceed to the oral glucose tolerance test. So OGTT test was performed at 7 and 40 days before the surgery. And the test involved administration of 40% glucose solution at 1.5 grams per kilograms body weight to the rats. Blood glucose levels were measured at intervals at 0, 10, 20, 30, 60, and 90 and 120 minutes post-glucose administration. We were using also a glucometer with tail clipping for blood sampling. It is important to know that the OGTT test was performed on each rat in the group. Group results were averaged and shown in following slides. Now we go further to these tables, which shows groups of rats selected of cafeteria diet on the right side, and on the left side is control diet group. Moving on the first chart, we see the differences in fasting glucose levels of 0, also 30, 60, 90, and 120 minutes after the glucose administration. And I have to highlight that the charts don't show standard deviation because of the clarity of the charts. Let me briefly now describe you what IT and sham surgery are. So ileal transposition is surgery that involves isolating and excising a segment of ileum, typically 25 from the ileocoelical valve, and transposing it to 5 to 10 centimeters distal to the ligament of Treis in the digestive tract. However, sham surgery is kind of the placebo surgery. So, well, let's move back to the general scheme of the experiment. After surgeries, these rats further divided into subgroups, each of them containing 6 rats in this subgroup. To clarify, this schematic refers to the entire experiment. However, only a part of the results will be shown later in the presentation. In the results, I will discuss only the blue and the green subgroups here. Okay, as you notice, I also added to some of the dyes the curcumine. The curcumine induces enzymes related to gluconeogenesis and which can positively affect further results. So having established the methods, let's now transition to the results section. So first, I will show 2 tables of 2 subgroups, both being on before and after sham surgery on control diet. I have to notice that the first 3 slides with tables are a comparison of subgroups with and without curcumine. So now take a look at the chart. We can see also the difference in fasting glucose levels also at the 10 and 20 minutes after glucose administration. And let's move to the another results. This table shows 2 subgroups, both on control diet before and after IT surgery. And now we can move to the chart. So at this chart, the only difference, as you can see, is in this place, is 120 minutes after glucose administration. Here we have another tables showing 2 groups, but before the surgery in these groups, these rats were first on cafeteria diet. After the IT surgery, they were on control diet, also with and without curcumine, as you see. Now we can on the chart. Here, there are also differences between only at the start of the OGTT test and at the end, at 120 minutes post-glucose administration. And let's move to the most relevant comparison. We compared 2 subgroups, both before and after control diet. One of them is after sham surgery, and the second one is after IT surgery. So moving on the chart results, because they are the most important. As you can see, we see the differences in fasting glucose levels at 10, 20, 30, 60 till 90 minutes post-glucose administration. After IT surgery, glucose level is lower than the sham surgery post-administration. And in addition, there is a small area under the curve, AUC, which indicates increased glucose tolerance in the IT group. So now to summarize these all charts. The main points that have been made are, so ILIA transposition surgery notably improved glucose tolerance. Also, curcumine has a beneficial effect on glucose tolerance. And these conclusions underscore the potential therapeutic benefits of iliotransposition surgery operation in managing glucose metabolism. And let me finish there. If you have any questions, you feel free.
Okay, thank you for your presentation. So at first, I'm not really into working with animals in laboratory. So I have a question about a diet, because you said the cafeteria diet is like a Western diet causing obesity, etc. So how the control diet look like? And second question is, of course, about your own contribution, especially the part of the surgery. You do it on your own or somebody help you during these steps?
First, I will tell you about the second, what you ask. I was taking care about this little brave patients, Sprague-Dawley rats. But how about this diet? I think it was without any sweets, like as you see at the first figure, without any Haribo or any biscuits. It's a normal diet, like a regular to the control diet. You want something to tell?
Okay, I'm just asking because I don't know how to work with animals, so thank you for your answer. And the second, about own contribution?
So I was taking care about these rats, and also I'm taking the results of OGTT tests. But also during this OGTT test, I also add this first picture that you see. Supply the glucose into these rats. So
okay, thank
also,
you.
In additionally, I create this kind of the presentation, and also I will help with creating the charts with these stars, and and I think that's enough.
Okay, great, thank you.
I have a question about the procedure of blood collection for the glucose test. Where place did you collect the sample?
We, as I mentioned, we take the blood from the tails of these rats.
Okay, thank you.
Of course, I have a question, but I wait if
you're waiting.
Thank you. First of all, I have some concerns about the thing you've mentioned, that some surgeries, yes, are quite safe, and they are safe and can be useful in the treatment of obesity, yes, and diabetes. But don't you think that it is the last step, and we should think about, I don't know, it is quite an aggressive type of, of treatment. Do we have any other methods just to, I don't know, help the obese and diabetes? Because doing a surgery, yes, it is for me the last step of treatment, yes.
Yes, I understand, but as I mentioned, the obesity has become an epidemic for a lot of people. So,
but it is. What are the reasons of the obesity? Our type of living, yes, our diet, yes, and things that we can change.
Not exactly, because we have a lot of metabolic disorders that prove to the obesity. Not only the style of your life results the obesity.
Yes, of course. And of course, sorry, I don't want to interrupt you.
So the obesity is a psychosomatic illness, so we have to focus not only on the diet, but for example on psychiatric help. And I want to mention about this surgeries. So the surgeries increase the incretins, for example GLP-1, which is comparable to the, for example, Ozempic,
no. Which is now widely popular, yes, as we know. But don't you think it is only my concern maybe that after the operation, after the surgery, people who are having some problems with food, yes, they will just come back to the point they were started before the surgery?
It is possible, but we have to remember about what I said before about this psychiatric help. And I think the diet, the psychiatric help, and the pharmacology or bariatric
all involved in treating the patients. And I just like to ask you about the rats. Firstly, I'm quite amazed that they were living even in 40 degrees cages. Really, I didn't know that rats are so fond of heat. But the other thing is, don't you think that rats, which are breed, yes, in some laboratory condition, could have the differences in level of glucose caused of stress? Because they were stressed, yes, we're taking the samples. It may higher the level of cortisol, for example, and influence the concentration of glucose, for example. And what's more, during the test which you've described, rats should not move. They should be still, because every activity will lower the level of glucose. And what do you think about it? How you just resolve this issue?
It can increase glucose tolerance, but I can say
as I remember, the stress was by the control. It was controlled during the operation. I don't know how about these cages, but I think they are raised from the baby rat to male adult rat. So I don't think so. It missed at something and have more stress about that.
I'm thinking not about the breeding condition, but about all this procedure, laboratory procedure that you've done to these rats, yes? Taking them from the cages, yes, taking the blood samples. It is not the physiological conditions of the rats, yes?
I think you don't know how to work with rats because...
I do know. I have some researches on rats, to be honest, yes. But I don't want to.
And they do everything, and they will become if they get the food. So.
But that is true, especially quite similar to humans, to be honest. I would like to mention that to your work, yes, that some procedure could higher the glucose level during the experiment, yes. Thank you. Thank you. I know that we have lack of time.
Bardzo dziękujemy za prezentację i za całą dyskusję. Teraz chwilka dla naszej komisji na wypełnienie kart ocen. I już w międzyczasie zapraszam autorów kolejnej pracy.
Zapraszam, to jest praca 10, prawda?
Tak, bo autora pracy 9 nie było, więc tutaj niestety.
Jasne. Chciałam się upewnić, dzięki.
Oczywiście. Zapraszam autorów kolejnej pracy. Analysis of the anatomy of the radial nerve and the arcade of flows in the cubital fossa using human cadavers.
Good morning.
Pani doktor, czy możemy już zaczynać kolejną prezentację?
Tak, jasne. Dziękuję.
Dziękuję.
Again, good morning, ladies and gentlemen. My name is Kamil Górecki, and I am a 4th-year student at the Silesian Medical University in Katowice. And I would like to present the findings of the study, analysis of the anatomy of the radial nerve and the arcade of Frosch in the cubital fossa using human cadavers, which we have conducted together with Radosław Karaś and Aleksandra Kepczyńska as the members of student scientific group at the Department of Anatomy. Special thanks are owed primarily to Dr. Tomasz Lepich for his assistance and supervision of our project. The radial nerve is a peripheral nerve that originates from the posterior cord of the branchial plexus and carries fibers from C5 to TH1. At the level of lateral epicondyle of the humerus, the radial nerve divides into the superficial branch of the radial nerve, exactly right here, and the deep branch of the radial nerve, exactly right here. Upon entering the arcade of Frosch, the deep branch of the radial nerve changes its name to the posterior interosseous nerve of antebrachy, exactly right here, and there is the arcade of Frosch. The radial nerve also contains the motor, sensory, and proprioceptive fibers. The arcade of Frosch was first described in 1908 by Frosch and Frankel. It is the highest proximal part of the supinator muscle, which can be either tendinous or membranous. A tendinous arcade of Frosch can serve as a site of compression of the extension of the deep branch of the radial nerve passing into posterior interosseous nerve. The purpose of our work was anatomical analysis of the division of the radial nerve into its terminal branches, along with measurements of the nerve division distance from the arcade of Frosch and the diameters of the radial nerve, its deep branch and superficial branch, as well as assessment of the nature and width of the arcade of Frosch. We used 8 upper limbs of cadavers for the study. We dissected the structures of the cubital fossa and made detailed measurements of the radial nerve. Okay. The average distance from the point of branching of the radial nerve into its terminal branches was 54.64 millimeters on average. In half of the cases, deep branch of the radial nerve divided before entering to the arcade of Frosch. The average distance from the point of branching of the deep branch of the radial nerve to the arcade of Frosch in these cases was 13.88 millimeters. That was that distance. Detailed measurement results are presented exactly right here in table number 1. The diameter of the radial nerve measured 10 millimeters proximally from the division site into terminal branches was 4.79 millimeters. The diameter of the deep branch of the radial nerve measured 10 millimeters distally of the site, of the origin site, averaged 4.08 millimeters, while the diameter of the superficial branch measured again 10 millimeters distally from the division site, averaged only 2.50 millimeters. Detailed results of diameter measurements of the radial nerve, its deep branch and superficial branch are presented exactly right here in table number 2. The width of the arcade of, the width of the arcade of Frosch averaged 8.60 millimeters. Additionally, 5 tendinous arcade of Frosch, 2 left upper limbs and 3 right upper limbs, and 3 membranous arcade of Frosch, 2 left upper limbs and only 1 upper limb were identified. Detailed results of the width measurement and assessment of arcade of Frosch structure are presented in table number 3. In our study, the tendinous arcade of Frosch was observed more frequently. Exactly like in other studies, we can see the results of other authors' studies, which they also presented that tendinous arcade of Frosch was more frequently. But we have to remember about one more important thing. We have to remember that the availability of human cadavers used for scientific research and medical student education is of course limited. Many medical schools face a shortage of human cadavers in the dissection room. This makes scientific research conducted on human cadavers valuable. A thorough understanding of anatomy of the radial nerve and its branches in the cubital fossa, as well as the arcade of Frosch, it is of course important for the development of anatomy and may also contribute to to the reduction of surgical complications during procedures in this area. That's the references. And at the end, as the authors, we would like to thank the people who donated their bodies to the science. The results of anatomical studies conducted on human cadavers can help improve the effectiveness of patients' treatment, of course, again, especially in the surgical procedures. Thank you very much for the opportunity to present our study here, and thank you very much for your time and your attention.
Bardzo dziękujemy za prezentację i zachęcam komisję do zadawania pytań.
Thank you for your presentation. It was really great. And I would like to ask you, of course, about your own contribution into researches and how your researches can be useful in real medical life. I'm really curious. So,
okay, so about my contribute, we together worked in the dissection room. We make the dissection of cubital fossa and the and made measure of the radial nerve and other structures. And of course, in the clinical usage, it is important because we got so much anatomical variation. So we have to know what to do during the procedures. So it's important for the future surgicals to make opportunity to train on something.
okay, thank you so much.
How the differences in anatomy could impact the weight of surgeries. I mean, how different anatomy, yes, can be, I don't know, a threat for surgical, yes. How can we help the patients with different anatomical parts?
Okay, I understand. It's very good questions because it's important because if the surgeon knows that some anatomical variations, some anatomical variations are common or more or less common, it is helpful for him to not make any mistake during the procedures. And that's important.
And for example, cut some muscles, yes, some nerves,
or
yes.
Some nerves, exactly, or the vessels.
Thank you, yes.
So it is important for all the surgeons so they could be careful during the procedures and they would not be surprised by anatomical variations of any nerve.
If they see something, yes, different from they were learned.
That is possible to look a bit different.
And you have only 8 upper limbs, yes, to work with, yes?
Exactly. Unfortunately, as my colleague mentioned, the cadavers are limited. So
yes,
unfortunately, we don't have much more of them, but we will try to enlarge our group.
So the conclusion of your work could be that we have to think how to, I don't know, how to get more, yes, material to work on. Yes. Thank you so much for it.
Thank you so much.
Very much.
Bardzo dziękujemy za całą prezentację i dyskusję. Teraz chwila dla naszej komisji na wypełnienie kart ocen i już w międzyczasie zapraszam autorów pracy The Relevance of Reperfusion Stroke Therapy for Extravascular MER175B Expression in Acute Stroke. Czy pani doktor już jest gotowa?
Tak, jestem gotowa, dziękuję.
Dziękuję.
Dear committee members, ladies and gentlemen, my name is Aleksandra Tobiasz and I'm here to present our study, The Relevance of Reperfusion Stroke Therapy for miR-17-5P expression in acute stroke. What is a stroke? Stroke is a focal neurological impairment of sudden onset, lasting more than 24 hours and of presumed vascular origin. It is one of major causes of death and disabilities in developed countries. Currently, intravenous thrombolysis, mechanical thrombectomy, or these two methods in combination are considered an effective treatment for stroke. However, mechanical thrombectomy has been unsuccessful in up to 30% of treated patients. That's why identifying factors limiting the clinical benefit of mechanical thrombectomy could improve therapeutic strategies and better guide stroke reperfusion treatment. In the age of emerging personalized medicine, biochemical and molecular biomarkers are being investigated to improve prognostic value with respect to the course of stroke and post-stroke disabilities. That's why we started looking into microRNA. They are small non-coding RNA molecules which have been found to control gene expression, primarily by silencing the gene. They are critical to cell physiology and development, and they are also implicated in pathological processes, such as autoimmune diseases, viral infections, and carcinogenesis. Our attention was particularly focused on miR-17-5P, since the research suggests that it may be involved in stroke pathophysiology. Some studies indicate that it can contribute to neuroinflammation, neuronal apoptosis, and blood-brain barrier dysfunction, which are key mechanisms in stroke progression and development. Also, a human study on miR-17-5P isolated from serum extracellular vesicles showed a significant level increase in patients with ischemic stroke compared to healthy controls. What's more, a significant upregulation of miR-17-5P was reported also in the serum, plasma, or PMBC in ischemic stroke patients compared to healthy controls. By examining patients' profile in the first hours and days after an ischemic stroke, one could predict which processes, neuroprotection or neurodegeneration, will predominate in that patient. That's why our study aimed to identify microRNAs as potential biomarkers for stroke diagnosis and prognosis. In this research, we included patients aged 50 to 85 years with first ever symptomatic ischemic stroke, pre-stroke status of 0 to 2 MRS, and without any other severe neurological disorders. Using this criteria, 72 patients were qualified into the study. Before I continue with my presentation, let me briefly explain what the MRS is. The Modified Rankin Scale is a scale used for measuring the degree of disability or dependence in daily activities of people who suffered a stroke or other causes of neurological disability. For outcome prediction in clinical trials, MRS is usually dichotomized. 0 to 2 points stand for good functional outcome, and 3 to 6 points mean poor functional outcome. And thanks to this scale, we can assess patient's condition. Going back to materials and methods, our patients were divided into 4 groups based on the method of treatment. First group received only aspirin. Second group underwent intravenous thrombolysis. Third group had mechanical thrombectomy, and the last group received combined therapy of intravenous thrombolysis and mechanical thrombectomy. On the first and 10th day after the stroke onset, blood samples were collected from each patient, serum was isolated, and the samples were stored until further analysis. Later, exosomes were isolated from serum. The QRT-PCR was performed to measure the expression of miR-17-5P, and the relative miRNA level expression was calculated. Statistical analysis was divided into 2 groups: analysis of differentially expressed miRNAs between first and 10th day from stroke onset, and it was performed within the groups and also between each treatment group. And the second part was analysis of differentially expressed miR-17-5P between patients with good and poor functional outcome. Let's move on to the results. Temporal analysis of miR-17-5P expression revealed that only in patients treated with intravenous thrombolysis and mechanical thrombectomy, there was a significant changed in mRNA level. It decreased between the days of admission and the day of discharge. Then we compared miR17.5P expression fold change between the groups, and we found that the expression level was significantly lower in the group treated with intravenous thrombolysis and mechanical thrombectomy compared to the group treated with intravenous thrombolysis only, but no other statistically significant differences were noted for the remaining treatment groups. And finally, we tested miR17.5P for its association with functional outcome, and we found that the expression was significantly upregulated in people with good functional outcome compared to patients with poor functional outcome on the day of discharge. Let's move on to the conclusions. Using patients' miRNA profiles when selecting them for certain types of reperfusion treatment may help to reduce the risk of failure and improve the clinical effects. Extravesicular level of miR17.5P can be linked with functional outcome. However, more research is necessary to further explore this topic. Thank you for your attention. If you have any questions, I will gladly answer them.
Thank you for your presentation. I have one question. What about the stability in serum of this particular miRNA? Because it will be for a diagnostic, so it will be stable from collecting to the testing.
In the center where we collected the samples, which was in Ohio, there are fridges for minus 80 degrees, and the samples were stored there.
So the sample was stored or the extracted miRNAs?
The extracted mRNAs were stored in the fridges to make sure they are still stable. Yeah.
Okay, thank you.
The question previously asked was crucial, yes, because when we are having patients who are suffering stroke, we have to be sure that the result, yes, is real, yes, not connected with the lack of the particle in the sample. I'd like to ask you, how long does it take to analyze the MIR, yes, in the test? You know, patient is coming to the hospital, yes, and we have to know in the very little time, short time, what is his state, his or her state, and how long does it take to do the results?
I think that
from taking the samples, yes, to the result.
Like right now, it wasn't our goal to do it as quick as possible. But in the future, it is actually, we think that it will be possible to take blood test from even a finger from patient and do it near his bed, because the methods of evaluating the miRNA could be similar to the ones that we have with COVID. Like, we don't need really any other special technology to evaluate the miRNA level, or we won't in the future.
So talking about COVID, you mean, for example, gene expert tests? Yes.
No, like PCRs.
Okay, but gene expert, yes, was the device used in COVID infections, but it works for, I don't know, an hour to get the results. And as you know, for sure, there is the golden hour, yes, in stroke when we can react. My other question is, you have patients from the age of 50, yes, to 85, yes? Is it correct? So do you have any idea if the level of your protein, your MER, could be different in elderly patients, for example? Because, as you said, these particular, type of MIR is, for example, connected with some brain injuries, brain inflammation, maybe it can be influenced by the age of the patients, not especially for the stroke.
But, mRNA are quite new molecules to test. We don't have any evaluation for what should be the right mRNA level in patients. So we don't know yet, but maybe it can be
Could be investigated, yes.
Yes, in the future.
And maybe the last question, do you know any other laboratory test which could be useful in stroke diagnosis? Okay.
I'm not sure.
It's okay.
Okay.
Thank you.
Thank you.
Thank you
for your questions.
Thank you.
Bardzo dziękujemy za prezentację i całą dyskusję. Teraz pozostawiamy czas dla naszej komisji na podanie kart ocen, a w międzyczasie już tutaj zapraszam na środek autora przedstawiającego pracę o tytule Immunogold Labeling as a Prospective Complementary Method to Immunohistochemistry of Biosection.
Jestem gotowa, dziękuję.
So, dear audience, my name is Maciej Baron. I'm representing the Department of Histology and Cell Pathology of Medical University of Silesia. So, So let's start with background. I would like to begin by showing you how today's medicine might be divided. There are two major models of medicine we know today. The first model is the evidence-based medicine. It is the most standard model of medicine we know today. It is determined by three major compartments. The first one is the doctor's clinical experience. The second one is the research evidence. And the third one is the patient's own preferences. However, on the other hand, we have a more futuristic approach. We have the personalized medicine. This medicine model evaluates the patient's organism individual. Okay, all right, can happen. So, individual's responses. Okay, so right now, let's compare these two medicine models. Let's start with the evidence-based medicine. This medicine model is based strictly on large studies and the statistics. And as you can see here, it evaluates only few parameters. However, on the other hand, the personalized medicine is based on the knowledge about the pathology and etiology of the disease. And as you can see here, it evaluates much more parameters. Moreover, it is dependent on the availability of the diagnostic procedures. So right now, let's talk about these diagnostic procedures, especially in personalized medicine, especially in cardiology. So historically, one of the first steps of personalization of medicine was the usage of transmission electron microscopy. Then the PCR has been invented, and today we have also the immunoelectron microscopy, the Katowice method. It is worth mentioning that all of these three methods are still being used in our department for the diagnostic procedures. But right now, there's a question. Do we really need them? Do we really need novel methods of tissue analyzing? Yes, we do. We need them to perform much more precise diagnostics, to be much more sure about the diagnosis. Okay, so let's carry on to the materials and methods. So we have used 6 endomyocardial biopsies from 6 different patients. We have used anti-human antibodies conjugated with a gold particle, especially anti-CD54, anti-von Willebrand's factor, and anti-human leukocyte antigen class 1. we have used cryo-ultramicrotome, which was dedicated to Tokuyasu method, and we have used transmission electron microscopy. So, how did we prepare the samples for the Tokuyasu method? The first step was to frozen the tissues by using a liquid nitrogen. Then the frozen tissues were cryo-sectioned on cryo-ultramicrotome. You can see this process right over here, and as you can imagine, it requires a pretty high skill. Then the section samples were immunolabeled using anti-human antibodies conjugated with a gold particle. This gold particle might be later seen under the transmission electron microscope as a black dot. Okay, so let's carry on to the results. So here, you can see the endomyocardial biopsy of one of the patients. We were diagnosing this patient for the myocarditis. Thus, we were immunolabeling von Willebrand's factor, which might be overexpressed during the inflammation. And, as I said before, every black dot is a point of the expression of this molecule. Okay, here you can see another immunolabeling, this time for human leukocyte antigen class 1. And right now, I have a question for you. Regarding on my prior words, can you see the place of expression of this molecule? I'm going to give you a moment. And here you can see four examples of it. So, finally, let's move on to our conclusions. So, firstly, why should we really use the Tokuyasu method? So, the first advantage is the fact about its high sensitivity, especially for intracellular proteins. Moreover, this method is able to detect specific biomarkers which are impossible to detect in any other methods. Moreover, this method has high specificity. However, despite its effectiveness, this method is not perfect and it has few disadvantages. The major one of them is the fact, as I said before, that the cryosectioning requires extremely high skill. Moreover, the method itself is pretty time-consuming, and usage of antibodies and gold particles makes it a bit expensive. However, we do believe that the development of science might lead us to overcome these obstacles in the future. So, thank you very much for your attention. If you have any questions, please feel free to ask. I'll be happy to answer them.
Okay, so thank you for presentation. At first, I'm really amazed by your energy on the stage. So the question is, of course, about your own contribution into the project. As you said, it's high skilled. So are you high skilled in your opinion?
That's a good question. Yes, I was preparing the samples, but I needed some help from professor to prepare this cryosection because these samples are really thin and they are pretty hard to put on the net to can be seen under the transmission electron microscope. So in that point, I needed help, and also I needed the help in the bioinformatics.
Yeah, but it's a training, yeah.
Yes, a bit.
For future.
Yes.
Okay, thank you.
My time?
Yes.
Yes. Thank you so much for the presentation. You are speaking so fast that I really have to focus on you to understand. No, it's okay. And I would like to ask you about how much will it cost and how long will it take to, obtain some results. But you've told us that it is quite an expensive method, yes, and time-consuming type of preparing the tissues. So how can it be used in the personal medicine? I mean, you gave us the examples of von Willebrand, yes, for example, yes, labeling, or other, yes, particle, I cannot remember. It was MGC classes, yes. Sorry. So how could it be used, yes, in the personal medicine?
Oh, and that's also a pretty interesting question because everything started with a professor who was a bit problematic with diagnose mitochondriopathy. So he decided to examine the patient even further, and he did the Tokuyasu method to know what exactly happened to this patient and why the mitochondriopathy is present on this patient. So he wanted to know the defective protein, and it was completely possible.
So it is mainly used for patients who are difficult to diagnose, yes?
Yes, exactly. Might be hard to diagnose, or if we aren't sure about the diagnosis. And what's interesting, it's not this method is not that horrifyingly long, because it's something about 1 week in our case, because we are.
Yes, it is not long. Yes, especially when we compare it to other methods, for example, connected with gene analysis. Yes, that can last for a longer time. And maybe it would be a controversial question, but do you think that artificial intelligence could be used in, for example, your method, the method you were describing? I mean,
also
for example,
an interesting question. I'm sorry for interrupting, but you hit the nail, because we are just trying to use the artificial intelligence to make the diagnosis faster as the first thing, and much more repeatable. And
yes,
we are trying. They are still trying. I was trying to put it into this presentation, but we didn't succeed in time with the artificial intelligence.
But I think it could be quite an interesting, yes, part of your work. Thank you so much.
Thank you very much.
Bardzo dziękujemy za prezentację i dyskusję. Teraz chwila dla naszej komisji i w międzyczasie zapraszam już ostatniego autora, ostatniej pracy o tytule Occurrence and Molecular Characteristic of C. perfringens strains isolated from patients with AAD.
Dziękuję.
Good morning, ladies and gentlemen. My name is Aleksandra Polańska, and I'm here to present the study that I conducted with my colleagues Martyna Gąsiorek and Jakub Pierożek. And that is the study about the occurrence and molecular characteristic of Clostridium perfringens strains isolated from patients with antibiotic-associated diarrhea of Clostridioides difficile etiology. Thank you. Okay, so to start, I'd like to say a few words about Clostridium perfringens. It's a bacterium that is gram-positive, anaerobic spore-forming, and it causes food poisoning, necrotizing enterocolitis, and also gas gangrene, which I think is the most known disease caused by this bacterium. And it produces many types of toxins, actually 14. But for our study, the relevant was alpha toxin, beta toxin, enterotoxin, iota toxin, and epsilon toxin. And to develop the topic of toxins produced by Clostridium perfringens, I'd like to mention alpha toxin. I think is the most well-known, and it causes intravascular chemolysis and coagulopathy in the mechanism of hydrolyzing membrane phospholipids into fatty acids. And that way it distracts the cell membrane. And the other one that is relevant for our study is enterotoxin, that causes food poisoning by binding to claudin family proteins and also distracting the cell. And the aim of our study was to isolate and identify Clostridium perfringens strains from stool samples of patients developing antibiotic-associated diarrhea of Clostridioides difficile etiology, as I already mentioned, and also to identify the toxin profile of the strains that we obtained. So at the beginning, we received 61 samples, stool samples from patients with AAD of Clostridioides difficile etiology, and we needed to prepare them. So at first, we collected 1 milliliter of stool from each sample, and then we subjected all samples to thermal shock. We warmed them up to 80 degrees Celsius and kept them in the temperature for 10 minutes.
And after cooling them down, we inoculated them on agar plates with 5% sheep blood. And we left the plates for 24 hours in anaerobic chamber. And we also performed aerobic control to be sure that there was no contamination in our samples. The following day, we isolated characteristic colonies with double zone of beta hemolysis, and we identified them by performing reverse camp test and also biochemical identification. After that, we froze all of the strains in microbanks for further tests, and the temperature at the freezer was -70 degrees Celsius. And for further tests, we already performed multiplex PCR to check the presence of toxin genes, and these were the genes for toxin alpha, beta, enterotoxin, iota toxin, and epsilon toxin. And here are our results. So we detected Clostridium perfringens strains in 18 of 61 samples that we tested, and that is nearly 30% of all the samples. And for the molecular tests, we only detected toxin alpha in all the strains. And the conclusions are that even though Clostridioides difficile still remains the main factor for nosocomial AAD, the Clostridium perfringens may also be significant. But interestingly, none of the strains that we tested had the enterotoxin gene considered to be responsible for gastrointestinal symptoms. So we can say that all the patients from whom we took the samples were only the carriers of Clostridium perfringens. And in the future, we plan to continue our study because there is a new problem appearing, and that is the emergence of Clostridium perfringens strains resistant to penicillin, which is the first-line drug against this bacterium. We want to assess the drug susceptibility of the strains that we tested. And that is all. Thank you so much for your attention, and we are open to a discussion.
Okay, so maybe I will start. Thank you, of course, for your presentation. It was really nice. And it's really great that you attached photos of you from laboratory. So I won't ask about own contribution because I think you do everything on your own. But I would like to know why Clostridium perfringens, why this bacteria you took it into your research?
So actually, our study is a part of a study conducted by the microbiology department. So we wanted to continue the study and use all the samples. So because we were inspired by our works tutor, we wanted to see if there's a possibility that Clostridium perfringens inhabits the intestinal tract of the person that has antibiotic-associated diarrhea of exactly Clostridioides difficile etiology. If that's possible, to have both bacteria in the same time.
Okay, thank you so much.
So my turn, I guess. So I'd like to ask you, who is prone to be infected by Clostridium? You told that it is connected with long antibiotics, yes, a treatment, but who else?
So, for example, patients with HIV or patients that take the drugs that can decrease their immune system opportunities. And also the patients that are elderly and are in hospitals for a long time.
I'm asking because my father is infected by Clostridium. He was in hospital for a long time, yes, and he was treated by antibiotics, and as well, he's taking some drugs which you've said are decreasing his immune ability. So I'd like to ask you, how should I live with my father? I mean, can me and my family members be infected by Clostridium living with my father using the same tools, for example?
Of course, that's possible. But if your immune system is okay and you don't take any drugs to that decrease its opportunities, I think that it is more possible to be a carrier of Clostridium difficile, and that is also relevant because then you need to be really careful around elderly people because you can transmit the bacterium to them.
Is there any way to get rid of it?
Not really. I think antibiotics, but we use antibiotics only for the patients that are hospitalized because we also need to be careful with them not to get too much resistant bacteria. So we use, for example, fidaxomicin or vancomycin for the hospitalized patients, but we don't use them for family members of the patients because it is more relevant to cure the patients that are already ill, that are not only carriers of Clostridium perfringens, but have the disease, have the diarrhea. So we basically can't use them for people that are just carriers.
I know I've heard that the doses of antibiotics which are necessary to get rid of Clostridium are pretty high, yes, and can affect, for example, I don't know, the kidney or the liver of the patients, yes? Is this true?
Yeah.
Thank you so much. Thank you.
Thank you.
Thank you very much.
Bardzo dziękujemy za prezentację. Teraz już po raz ostatni proszę o uzupełnienie kart ocen. Ze względu na to, że od uczestniczki, która nie prezentowała dzisiaj pracy, a była wypisana na listach, dostałam informację o tym, że nie dotrze do nas. Z tego względu ta praca nie będzie dzisiaj zaprezentowana.
W porządku.
Więc doszliśmy do momentu, gdzie wszystkie prace zostały zaprezentowane. Ze swojej strony gratuluję Wam ogromnie wszystkim, ponieważ to niesamowite, że chcieliście tutaj dzisiaj z nami być i zaprezentować wyniki swojej ciężkiej pracy. W tym momencie chciałabym też bardzo poprosić Panią doktor o zabranie kilku słów na zakończenie naszej sesji.
Szanowni Państwo, ja chciałabym podziękować wszystkim Wam i każdemu z osobna za niesamowite zaangażowanie i odwagę w prezentacji dzisiejszych wystąpień. Ja naprawdę jestem zaszczycona, że mogłam uczestniczyć w tej sesji, dlatego że uważam, że poziom Państwa wystąpień był naprawdę wysoki. Robicie Państwo wspaniałe rzeczy i mocno trzymam kciuki, żebyście mogli kontynuować swoją pracę naukową, żebyście mieli z niej satysfakcję i osiągnęli wyniki, które będą Was satysfakcjonować. Jestem szczerze wdzięczna innym członkom komisji za poświęcony czas, za bardzo trafne pytania i mam nadzieję takie wsparcie merytoryczne uczestników dzisiejszej sesji. No i oczywiście, Szanowni Państwo, wielkie ukłony dla organizatorów, dlatego że domyślam się, że logistyka tego wydarzenia była dość interesująca, więc jesteśmy wdzięczni, że mogliśmy dzisiaj w tej konferencji uczestniczyć. Jeszcze raz serdecznie Państwu gratuluję, podziwiam odwagę, podziwiam Państwa umiejętności językowe, zaangażowanie i składam ukłony przed Waszymi opiekunami. Naprawdę uważam, że wykonaliście Państwo świetną robotę. Dziękuję.
Bardzo dziękuję Pani doktor. W takim razie sesję nauk podstawowych, biotechnologii i inżynierii biomedycznej uważam za zamkniętą. Serdecznie Was jeszcze tutaj zapraszam na warsztaty, które odbędą się tutaj w tym budynku od godziny 12. Te, na które były zapisy, ale jeżeli będą wolne miejsca, to można mimo wszystko w nich uczestniczyć. I też tutaj na korytarzach będą stanowiska z różnymi takimi warsztatami, na które nie było zapisów. Też o godzinie 12 rozpoczyna się lunch w holu przy auli A3. To jest ten budynek, w którym Państwo się rejestrowali, czyli jest to ulica Medyków 18. Jeszcze raz Wam wszystkim niezwykle dziękuję. Bardzo jeszcze raz dziękuję członkom komisji, tym stacjonarnym, jak i Pani doktor, która była dzisiaj z nami zdalnie. I mam nadzieję, może do zobaczenia za rok.
Idźcie i jedzcie. Do zobaczenia.